Assalamu alaikum, everyone. Today, inshallah, we're going to continue with the techniques that are used to study gene expression. In the previous lecture, we learned about reverse transcription PCR. We learned about microarray. Another way of detecting gene expression is through the use of an approach called RNA sequencing or RNA-seq. So the principle of this technique relies on the isolation of all mRNAs. So this technique could be used to study the expression of all genes inside the cell. Remember that if we're talking about eukaryotic cell, so this is the nucleus. In eukaryotic cells, all the genes that are expressed will have mRNA in the cytoplasm of the cell. So these mRNAs are extracted from the cytoplasm or the cytosol. Okay, so any gene that is expressed will have an mRNA inside in the cytosol or in the cytoplasm region. So this mRNA will be isolated, or all the mRNA. Again, this is all the mRNAs. If a gene is expressed, we will find an mRNA for that gene, corresponding to that gene. And now, these mRNA molecules, of course, there will be billions of mRNA molecules or millions of mRNA molecules isolated from maybe hundreds of thousands of cells. And then the mRNA molecules are cut into smaller fragments. And then these fragments are sequenced. So the fragments first are converted into cDNAs, okay, using reverse transcription PCR. And then these small fragments are sequenced. And the sequenced small fragments will be mapped to a genome sequence. So this step is called mapping. Mapping means we want to determine the location of each sequenced fragment with respect to the genome. Okay, so we know the genome sequence. So for example, here we have this fragment has a sequence that is located here within the genome. This one is located here. So these are basically, these are sequenced cDNA molecules, which corresponds to sequenced mRNA molecules. Okay? Sequenced mRNAs. So in terms of mapping, so mapping sequenced mRNA molecules. Basically, we determined the sequence of mRNA molecules, sequenced mRNA fragments, because so if we have, for example, part of the gene molecule, okay, so this is just a region of the genome. And this is just, I'm showing you part of the genome. And we have let's say here we have a gene consisting of 3 exons. So here we have, let's say, gene X, and here we have, use a different color, gene Y, gene Y, consisting of 4 exons. So this is exon 1, exon 2, exon 3, exon 4. And likewise, here we have exon 1, exon 2, exon 3. And lastly, we have a gene maybe consisting of 2 exons like this. So this is gene Z, exon 1, exon 2. Okay. So following sequencing, so we will have, let's say, millions of sequenced cDNA molecules. Or billions of cDNA sequences. Again, they represent the fragments that are made from the mRNA fragments, and the mRNA fragments are made from whole mRNAs. So in mapping, what happens is that some of these RNA fragments will map to expressed regions like this. Okay, so the next step is to do something called mapping, which is used to determine the location of the millions of cDNA sequences with respect to the genome. Okay. So after mapping, for example, so here, these are cDNA molecules, cDNA sequences. So each line represents a cDNA sequence mapped to a genome, okay? So all these cDNA molecules, their sequences are identical to sequences in exon number 3 of gene X. All these cDNA molecules, they have sequences that are identical to sequences within exon 2, similarly here, 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 here. So after doing mapping, we could determine if gene X is expressed. We could also determine the level of expression. For example, if gene X, of course, here we're trying to answer yes or no. We could also answer the level of expression, high, low. We could also determine the alternative, or the what we call the splice variant. Is it consisting of exon, for example, exon 1. Okay. Let me add here exon 4, exon 2, exon 3, or exon 4. So after we do mapping, after we determine where does every sequence of cDNA map to the genome, we can answer for each gene these three, we can gain these three pieces of information about the expression of each gene. So for example, by judging from the mapping, so because we have cDNAs, cDNA molecules, or cDNA sequences mapping to the exons of gene X, this means that we already isolated mRNA for gene X. So this is mRNA X, and now mRNA X was isolated, and then mRNA X, okay, was fragmented and then sequenced. Or maybe it's before sequencing, cDNA must be made. And now we have cDNA molecules from mRNA X. And these cDNA molecules, okay, so we could say mRNA X cDNA molecules, cDNA fragments. Okay? So, of course, we have other fragments from other mRNAs, and we have cDNA molecules from other mRNAs. Okay? But here we're just focusing on mRNAs. So if we go backward, the mapping of cDNA sequences to the gene means that gene X was expressed. How did I know? Because these cDNA molecules, these sequences were obtained from cDNA molecules, and these cDNA molecules or cDNA fragments were obtained from fragments of the mRNA X, and these fragments of mRNA X were obtained from after isolation of mRNA X, and this mRNA X was isolated from the cell, which means there is an mRNA X corresponding to gene X in the cytoplasm, which means the gene X is, which means that gene X is expressed. So, gene X is expressed. Yes. How did I know? Some cDNAs are mapped to exons of gene X. Notice gene Y is expressed. We can ask the same question for gene Y. What do you think about gene Y? Is it expressed or not? Gene Y. Yes, because we can see some cDNA sequences are mapped to exons within gene X. So the same answer for gene X. What about gene Z? What about gene Z? Is it expressed or not? No. No, none of the billions of cDNA molecules, their sequences were mapped to the DNA sequence of exons from gene Z. Okay? So gene X is expressed, gene Y is expressed, gene Z is not expressed. What about the level of expression? So we can so because these two only the genes are expressed. Notice that more cDNA molecules are or more cDNA sequences are mapped to the exons of gene X compared to Y. So the level of expression is considered high. The level of expression gene Y is considered low. What about the splice variant? Notice that only we have cDNA molecules that are mapped to exon 1. Sorry, here, let me change it like this. Oh. We have cDNA sequences mapped to exon 1, 2, 4, but not 3, which means in this type of cells, maybe let's say these are liver cells. So maybe we took the DNA or the mRNA from liver tissues. In liver tissues, gene X is expressed, but only includes exon 1, 2, and 4 in the final transcript. For Y, 1, 3, 4. Okay? So the mRNA of gene X in liver cells contains coding sequences from exon 1, 2, and 4, but not 3. For gene Y, exon 1, 3, and 4. Of course, Z is not expressed. Okay? This is how we read the result or the data from the RNA-seq experiment. Okay? Of course, using this technique, we can measure the expression of thousands of genes at the same time. Sometimes we want to use techniques that will tell us something about the gene function. Sometimes, or scientists are always trying to understand what is the function of a gene, or they try always to have some information about the gene function. One way to determine the gene function is to disable the gene and observe the consequences. So remember that a gene, okay, which sometimes we call a genotype, have a certain genotype, the gene will encode a protein or proteins, and this protein, along maybe with other proteins, it might carry by itself, let's say, a function, or with other proteins it will carry out this function. Now we have a function, and then we have a phenotype. So this function of this protein will produce a phenotype. So if we have a normal gene, we have a normal protein, normal function, and we have a wild type phenotype. Okay? So normal gene or wild type gene. We could, for example, determine the function of a protein by disabling it. Disable means تخريب. Okay? We could completely delete the gene or introduce mutations. So the idea is that we could remove the gene, okay? Remove the gene from cells. This means, let me use a different color. So we could remove the gene, so no gene, which means no protein, and maybe now we have, because there's no protein, Now, we might have no function carried out by this protein, okay? Or we could have a mutant protein, mutant gene. So we introduce mutations, okay? And then we have a mutant protein. It's not called a mutant protein. It's called abnormal protein. And then we could have abnormal function, abnormal or no function. And both could lead to what we call mutant phenotype. So the idea here is that if you disable a gene by removing the gene completely from the genome, or maybe make the gene a mutant by introducing mutation, you will end up with mutant phenotype. And this is what we do. We observe أو بنلاحظ this mutant phenotype in order to have some information about the function of the gene. Because if you remove the gene or disable the gene or destroy the gene completely, or maybe disrupt the gene or make it a mutant, you will end up with a mutant phenotype. And from the mutant phenotype, maybe the gene is important for your brain and maybe, or the brain of an organism. And if you remove the gene or disable the gene, there will be some problems with brain development, okay? Just like, and this approach, for example, was used to determine the genes that are important for development in Drosophila, if you remember in the previous chapter. So by following this approach, we can have some information about the gene function. Okay? Now, how can we disable genes or make mutant genes? We can make mutant genes by a technique called in vitro mutagenesis. In vitro means in a test tube. So basically, you have the gene in a test tube and you introduce mutations, and these mutations will change the protein or destroy its function. Okay? So the idea is that you have cells, and these cells have the gene. For example, this is the gene. The gene to be studied. Okay? And this gene could be isolated, okay, and could be cloned, just like we learned in this chapter. We could clone it into a plasmid, and then we could introduce mutations into this plasmid, into this gene. So we could do here in vitro mutagenesis. So this is one application. This is one use for cloning, is to help with in vitro mutagenesis. So this is, for example, a wild type or normal gene with a normal DNA sequence. And this is a mutant gene with mutations. in its sequence. Okay. Of course, how this technique introducing mutations, you will learn about this, inshallah, in other courses. But for now, we could take this gene and put it back inside cells. And now these cells will have the mutant version of this gene. Okay. And here we have normal cells. And now these cells with the mutant gene, they have a mutant phenotype. All what we have to do is to determine what is the function of this mutant phenotype. Okay, maybe the cell is unable to synthesize a particular protein, and this is why the cell is unable to carry out a specific function or a specific chemical reaction, or maybe something happened to the structure of the cell or the appearance of the cell, how the cell looks like, or maybe the cell is unable to synthesize a certain molecule or carry out a specific function. Okay? So we can edit genes using in vitro mutagenesis. Okay? Another way of editing genes is by using what we call the CRISPR-Cas system. This CRISPR-Cas system was adopted from bacteria, so it's a system naturally found in bacteria, and it is used by the bacteria to protect itself against viruses. But currently, scientists are using this CRISPR-Cas9 system as a technique for gene editing in living cells. Okay, so here we have in vitro mutagenesis, which means you do not edit, or you do not change the sequence of the gene while the gene is inside the cell. No, you do it in the lab, in vitro. So you put this, you clone your gene and you put your gene in a test tube and you do the editing inside the test tube. Okay? Then you introduce the mutant gene into live cells. Edit them to do what? You're just editing the gene. You are introducing mutations because you want to change, you want to destroy the gene, or you want to make the gene non-functional so that it will produce a mutant phenotype. And from the mutant phenotype, you might learn something about the function of the gene. Okay? The CRISPR-Cas system could be used to edit genes or introduce mutations into genes while the gene is inside living cells. So you don't have to take the gene out and do cloning and then use in vitro mutagenesis. How? The CRISPR-Cas system uses a protein and an mRNA, an RNA molecule. This protein is called Cas9, and this Cas9 is a nuclease. A nuclease is a protein or an enzyme that can cut DNA. So it can cut DNA, but it doesn't know where to cut the DNA. That's why it needs help from the RNA molecule. The RNA molecule is called guide RNA because it will guide the nuclease and it will tell the nuclease where to cut the DNA. This guide RNA consists of two parts. This part here will allow the binding of the RNA to the nuclease, and this part here is complementary to the sequence we want to cut. Okay, so this will be complementary to a sequence that binds to a target gene that we want to destroy, for example. So now we have the Cas9 guide RNA complex. The Cas RNA complex will bind to a specific DNA sequence. Maybe this is part of the target gene we are trying to destroy. because the guide RNA will be complementary to one of the strands of the gene. And after the guide RNA binds to its target sequence, the nuclease will cut the target gene. So here we have what we call, this is called double strand break, or DSP for short. Okay? So now, what happens next depends on on many parameters. But you have to understand, the CRISPR-Cas system only introduces DSPs to the DNA. What happens next has nothing to do with the Cas9. So when the DNA is cut, there are two, you have to understand, inside the cell, the DNA should not be cut like this. Okay? So what will happen is that once the DNA is cut, the DNA repair systems. So the DNA repair systems are proteins and enzymes that will try to fix this damage. So this is, here we have a gap. Gap يعني فراغ. And this gap represents damaged DNA. So this damaged DNA, or this gap, must be fixed. Okay? It cannot remain like this. So the DNA repair system will recognize this gap or this damaged DNA, and it will try to fix it. How? So we have a DNA repair system called, a mechanism called non-homologous end joining. Okay? So one way to repair the DNA is through a mechanism called non-homologous end joining. I think the O is before the O. Or N H G J, oh sorry, E J. In non-homologous end joining, what happens is that some nucleotides might be removed from here, and then the DNA will be joined together, or some nucleotides will be added. So here we have addition or deletion. Then we have ligation. So either some nucleotides will be removed from here and here, then the two ends of the DNA will be joined together like this, or some nucleotides will be used to fill the gap. So there will be added nucleotides and the two ends will be joined. Whatever the case, the gene, we have random nucleotides in this gene, and this causes disruption of the gene or disabling of the gene. So now we have something called knockout. Knockout means we have the gene is no longer functional. Knockout means, this is a term used in boxing في يعني الضربة القاضية في الملاكمة يستخدموها knockout. So now we have a mutant gene. Why? Because some nucleotides are removed from the gene, or some nucleotides are added to the sequence of the gene. In both cases, we have a knockout or a gene that is no longer functional because its gene sequence is changed or altered. Or if this damage happened, we have another mechanism. So this is one mechanism of repair. The other mechanism is called homology-directed repair. Homology-directed repair, or HDR. In homology-directed repair, a normal copy of the gene must be present and could be used as a template for the repair. So some repair enzymes or repair proteins will take this template and use it and learn about the sequence here and use it to repair the gene. So now we have normal nucleotide, and now the gene is normal. So this way, so for example, if we have a gene, so this way could be used to repair a mutant gene. So imagine if this is a mutant gene, and this is a mutant gene because here we have a G and a C. So in the normal gene, instead of having G here, so this is nucleotide number 10, for example. This is nucleotide number 10. Instead of having G and C, here we have C and T. C and G, or maybe let's do A and T. So this is a mutant gene, and we want to fix it. Okay, here we have, let's say, a G. So this is nucleotide number 10 of the gene, G and C. We cut it. Now we have G and C. After we cut it, the DNA with CRISPR-Cas, a normal copy of the gene that have the normal nucleotides, A and T, okay, will be provided so that this G and C will be removed and A and T will be added. And now this is nucleotide number 10. And now we have a normal gene. Okay? So as you can see, one of the applications of CRISPR-Cas is to make mutant genes, or maybe to fix mutant genes. Other methods of studying gene function is to use RNA interference. We learned about RNA interference and small interfering RNA in the previous chapter. So RNA interference could be used to silence genes, to prevent the expression of genes. And RNA interference, or RNAi, uses microRNAs or small interfering RNAs, and these small interfering RNAs, they block the translation of mRNAs. Or they destroy mRNA. Okay? So small interfering RNAs or microRNAs, they could be used to block the translation of mRNA or destroy the mRNA, completely destroy the mRNA. If the mRNA is not translated or degraded or destroyed, the gene is not expressed. So one way to study gene function is to prevent its expression. Okay? Because remember, let me go back here. An mRNA, a gene, a normal gene, will be converted into a protein by first being, so the gene is transcribed into mRNA, then the mRNA is used to make a normal protein. So we can also generate a mutant phenotype by using this strategy. So normal gene, no protein. So how can we prevent, and which means no function, which leads to a mutant phenotype. So the idea here is that this normal gene will give an mRNA, but we could use small interfering RNA, or we could use RNAi to prevent the translation from RNA into a protein, so there will be no protein. Okay? So the gene is normal. Nothing happened to the gene. The gene is normal. Nothing happened to the gene. Okay? So these are three ways by which we can generate a mutant phenotype, and by looking at the mutant phenotype, we could have some information about the gene function. Another topic discussed in this chapter is the genome-wide association studies. So you have to understand, we cannot use RNA interference or CRISPR-Cas or in vitro mutagenesis to learn or to determine the function of genes in humans, because it will be considered unethical. Okay? We could do these, we could use these techniques to, to determine the gene functions in other organisms, like in mice and monkeys and in Drosophila. But in humans, we cannot do this. So we can analyze, we need to use an alternative approach to analyze the genomes of a large number of people in order to find, in order to find some mutant phenotypes in some individuals. And from these mutant phenotypes, we could determine the gene that is responsible for the mutant phenotype. And from the mutant phenotype found in humans, of course, the mutant phenotype will lead to disease, some in humans. So in humans, we have what we call genetic variations. Okay, تنوع جيني. And this genetic variation might lead to phenotypes, mutant phenotypes. Okay, and this mutant phenotypes might lead to disease. So if we analyze the genome of certain people with certain diseases, we could, because they have mutant phenotypes, we could basically determine what type of genetic variation they have compared to normal individuals. So the idea is that these mutant phenotypes or diseases are associated with certain genes that are not normal, or maybe genes that are destroyed, or genes that are mutant. Okay? So genetic variation means we have, let's say, some caused by mutations. So some mutations might affect genes, and these genes, they are important. They will produce mutant phenotypes. They become mutant, and the mutant phenotype might lead to a disease, a certain type of disease. Okay? So we have something called genome-wide association studies. Genome-wide, we are looking at the whole genome, looking for genetic markers. Genetic markers are basically, they will tell us about the genetic variation. Okay? Genetic markers, these are changes in nucleotide sequences between humans. And these genetic markers, some of these genetic markers are found in people with genetic diseases, or people who have genetic diseases, or people who might have some mutant phenotypes. Okay. So an example of genetic markers are single nucleotide polymorphisms, or for short, they are called SNPs. Okay. Let me stop here. So SNPs are responsible for some of the genetic variation observed among individuals. So humans are different from each other because they have different nucleotide sequences, and some of these differences are actually SNPs, single nucleotide polymorphisms. And these SNPs are considered useful genetic markers. Genetic markers means these are genetic variations that could be used because they are different in different individuals. They could be used to detect basically certain medical conditions or certain diseases in humans. So SNPs, again, they are single nucleotide polymorphisms, which means we have substitution of single nucleotides at a specific location within the gene. For example, if we have this region of the genome, let's say, okay, and this region of the genome, for example, contains a gene called gene X, okay, and this gene X basically is maybe from here to here, gene X, and this gene X has a sequence, the following sequence, just part of the sequence. So both strands. Okay. And then here, this location, we have a location of a SNP. Okay. Which means if we analyze the sequence of gene X in, let's say, in a number of individuals or people, they might have different nucleotides at this location within the gene, only this location. So if we look at the genome of another person, so maybe this is a genome from an individual called Ali, maybe, and this is the genome from the same genome. So here we have gene X again. Okay, and if we zoom in to the same region, okay, this is the genome of, let's say, another person, maybe Sarah. And if we look at the sequence, if we read the sequence here, we have similarly G, T, A, T, C, T, T, C, C. Okay, so this is the SNP. So for both Ali and Sarah, every nucleotide within gene X will be the same except for this SNP, maybe. Okay, so now here the SNP, let's say this is location, the location of this is nucleotide 11,250 on the chromosome. Here, something like this. Okay, so maybe this is, let's call this chromosome 1. This is chromosome 1. Okay. At this location within the chromosome, we have a SNP, and this SNP is AT or CG. So some people will have AT or CG. Okay. This is not a mutation because Sarah is normal, Ali is normal. Okay, this is single nucleotide polymorphism. This is not a mutation. It's not affecting the function of gene X. Maybe it's an intron. So actually, the majority of nucleotide or SNPs. So some SNPs in coding sequences or in coding regions, like within exons, and some are in non-coding regions. Actually, not some. The majority of SNPs, they are in non-coding regions. Okay, but still, these non-coding regions, like they could be within introns, or they could be within regions of promoters outside the genes or enhancers, which means they could still have an effect on the expression, the level of expression of the gene. Okay, and within the coding region, these SNPs might be synonymous, or they might basically cause a silent change or missense change, or sometimes nonsense change. But most of the SNPs are found in non-coding sequences. Okay. Some SNPs are frequently found associated متعلقة with a particular disorder or a particular gene. Okay. Typically, they are not directly involved in so they don't cause, let's say, mutation in genes, but they might be associated with genes. How? So, for example, here we have a normal gene, allele. Let's say that this is gene X. Let's call it gene X. And this is the same gene X, but it's a mutant allele of gene X. Okay. So this mutant allele of gene X causes disease. This is the normal allele. Some SNPs, certain types of SNPs. So this is, let's say, this is a SNP location, SNP locus. Locus means location. This is the same SNP locus. And this SNP, which is maybe found, let's say that this is part of chromosome 2. Okay, this is chromosome 2, of course. And the location of this SNP within chromosome 2 is 275,030. So this is the location of the same SNP, 275,030. And this SNP, so SNP located as nucleotide number 275,030, it could be either A, T, or CG. Some people, they have AT or CG. But scientists found out that A and T, when an individual have an A and T on this locus, this individual will have a normal copy of allele gene X. Individuals that have CG at this SNP location, they will have the disease allele of gene X. Notice that this SNP does not cause a disease, or does not cause the disease. The disease allele of gene X causes the disease. So we say that this SNP is linked to a normal allele, the AT, and this SNP is linked to the disease allele. Okay? So all what we have to do, if someone has a disease, if we want to know if someone has a disease caused by gene X, all we have to do is look at the SNP. If they have AT, they probably have the normal allele and the disease is not caused by this gene. If they have CG, they might have their mutant allele and maybe the disease is caused by this disease-associated allele. Okay? And notice that this is the SNP locus is basically in non-coding region. Maybe it's outside the gene, maybe it's within the gene in the intron, or maybe so some SNPs, they are they might basically, so you have to understand that there are around so more than 335 million SNPs or SNPs found in all humans from all populations. Okay? So within the human genome, there will be approximately 4 to 5 million million sites, different between your genome, like any genome, like my genome, your genome, and the reference human genome. Human genome, this is the human genome that have been sequenced, okay, or a typical human genome we use for comparison. So basically, each one of us will have 4 to 5 million sites or loci, جمع locus, that are different. And these And 90, so basically we are different in 4 to 5 million location within our genome. So if we analyze the genome from all students, most of the genome will be identical except for 4 to 5 million sites. Remember, the human genome, the haploid human genome, consists of approximately 3 billion bases. Okay? So out of 3 billion bases, we have 4 to 5 million differences. And most of these 4 or 5 million sites that are different among humans, more than 99%, they are SNPs and small deletions or insertions. So basically, our genome, so most of these differences, okay, and these differences, by the way, are responsible for the genetic variation observed among individuals. Okay, so most of these differences are because of SNPs and small deletions and insertions. So why is it important to study these SNPs? Because these SNPs can tell us about, they can affect how humans, so SNPs, that can affect how we develop disease. So some SNPs have been associated with certain diseases or the risk of having certain diseases. Some SNPs can affect how we as humans respond to, I'm going to list a couple of things. Pathogens, chemicals, drugs, vaccines. So certain types of SNPs will make different people respond differently to pathogens like bacteria and viruses. So some people are more easily infected with viruses or bacteria because they have certain SNPs in their genome. Similarly, some people, they might respond to drugs in different ways because they have certain types of SNPs. So these SNPs is responsible for these different responses to pathogens, chemicals, drugs, and vaccines. We all know that some people, when they take certain drug, they will respond to that drug or they will benefit from that drug. But some other people, they might take the same drug and they will not get any benefit from the drug, or they will not respond. They will not have a response from that drug because simply they have certain types of SNPs. So these SNPs allows us to detect certain genetic diseases. They might be associated with certain diseases. They might be associated with risk to certain diseases, and they also might affect how our bodies will respond if we are infected with pathogens, عوامل ممرضة, or if some chemicals are inside our bodies, or whenever we take drugs or vaccines, لقاحات. Okay? The last concept in this chapter is about cloning organisms and cells. So we've learned about, so cloning is basically, as I defined it at the beginning of this chapter, is making more copies of a molecule or let's say something, making more copies of something. And we discussed DNA cloning, or more specifically gene cloning. And we could also use something called cell cloning. So in cell cloning, we can make more cells of the same cell, and we have also organismal cloning. We can clone a whole organism that is genetically identical to their parent, to the parent, actually one parent, not two parents. So cell cloning is done using types of cloning of cells called stem cells, خلايا جذعية. Okay. And we're going to discuss cloning of organisms also. So stem cells are unspecialized cells that can produce itself indefinitely, which means it can continuously divide, not continuously, they have the capacity to always divide to replenish itself, and it can also differentiate into more than one type of specialized cells. Okay? So there are three characteristics of stem cells. They are unspecialized, they can produce itself indefinitely. This is the other characteristic, خاصية مميزات, and can differentiate into one or more types of specialized cells. This is the third characteristic, أو الخاصية الثالثة. All right, so let me stop here, and inshallah we're going to continue tomorrow.